Okay, this is going to be a very short section, really. I taught a lot more about it before, but there's a lot of information on this online or anywhere if you get into it. And you're going to get into it if you're about permitting for point source pollution. And point source pollution happens to have a lot of organic material in it. And that's this whole notion of BOD, or biochemical oxygen demand. So what is that? I mean, what's that look like? We've talked about stuff before, but what's biochemical oxygen demand? I'm going to go BOD, biochemical. And really, if you look at well, let's have a look at it logically from the words. What would you kind of infer from the words over here on BOD? Take a wild guess over here.
Repeat the question.
What would you infer from these words, BOD, biochemical oxygen demand? I mean, what does that actually mean?
It's kind of like a general need for oxygen in the environment.
Okay. So it's the potential for whatever this thing is to consume oxygen. And in this case, biochemical oxygen demand is a catch-all for all oxygen that is required to decay an organic, something that's based on organic material. So the classic case would be effluent from a sewage plant. So we have effluent from a sewage plant. The majority of that might be carbon-based because, we've got toilet paper and feces and, detritus and sticks and stones and that kind of stuff. So there's a lot of carbon, but there's also nitrogen associated with it and phosphorus and potassium and everything else. So think about sort of effluent from a sewage plant. It's going to be, bits and pieces that are, of various sizes. Some of them are very, very small. But that eventually they have to get broken down. How do they get broken down? And we talked about this earlier. How do dead bits of plants and animals and toilet paper and that stuff, how does that get broken down in the water?
Bacteria?
Yeah, you've basically got bacteria that are essentially, their whole job is to decompose things. Imagine what would happen if we didn't have decomposing bacteria. For instance, we would be walking on miles of leaves. Well, of course, the trees would have died by now, but imagine if there were no decomposing bacteria and all of our leaves every year that came down onto the forest floor or something like that were not decomposed. Well, eventually they'd rise to being a lot higher than those plants. So decomposition, we need to understand, is a natural process. And so what are the energy sources that these bacteria, where's the energy coming from with these decomposing bacteria?
Respiration?
Well, they need to respire, but where's the energy coming from? It's that effluent or that biological, that sludge or whatever, okay? Because it's got carbon, nitrogen, oxygen, potassium, sulfur. So the bacteria are getting their energy. Let's say it's that sewage effluent sludge. The bacteria get their energy to live on that. So what's happening is you've got let's draw that bacteria, and he's eating the sludge. And for argument's sake, let's just keep it, we can think of sludges like sewage sludge or maybe sludge that comes from a pulp and paper mill, lots and lots of carbon over there. But these bacteria are going to go ahead and eat all of these biologically based particles, or biologically based particles, or whatever they would be. He'd eat them up. What happens? He gets energy from that, so it goes from ADP to ATP, okay? And these bacteria populations are built up, but to break down all of these things, to turn them into, among other things, CO2 and other bits and pieces, he needs oxygen to breathe. So he has to consume oxygen to make that breakdown happen. And so with BOD, this catch-all, what we're saying is sometimes all you want to do, you don't really care whether this is sewage sludge, you don't care if it's sludge from a pulp and paper mill, you don't care if it's sludge from a municipal waste site where, it's just dead yard waste or something. You don't really care. All you're interested in is its potential to consume oxygen. And this is biochemical, so it's mainly a biochemical process. The substrate there is going to be typically biological in nature, and what you can infer from that is going to be the dead, the bodies and the cytoplasm of dead plants and some dead animals. Another place, BOD, would be a slaughterhouse where you have a lot of blood, or so you've got animal material that has a potential to consume oxygen. Does that make sense? So BOD is this whole idea of how much oxygen does it consume, and we don't care what the actual substrate looks like. We just want to know, from this plant, we can basically measure the potential for oxygen consumption. Okay? So that's kind of in concept. If you have a look, BOD, you've got your organic waste plus your O2, and you've got your little bacteria munching on there. And those bacteria then produce carbon dioxide, water, nitrates, phosphates, and a whole lot of other stable products which are going to go out from that. So that's kind of, it's a metabolic process. These guys are useful to us. That's what we use in sewage plants in any case, okay? So that's the basic process over there, and we're always interested in how much there is. Now, there are bunches, but for the purpose of this class, we're looking at really two kinds of biochemical oxygen demand. And the one is carbonaceous BOD, and the other one is NBOD. CBOD and NBOD. CBOD plus NBOD will basically end up the total biochemical oxygen demand. Now, the difference between the difference between CBOD and NBOD is that carbonaceous biochemical oxygen demand, because carbon tends to be, a lot of your carbon molecules will be fairly large and complex. So that process, the CBOD process, is often, if you had to look at it on a chart against time, it would kind of look stable. You'd think I'd learn. That decay process would sort of look stable. So over time, total O2 consumed might look like this over time, and that time might be, say, 90 days. Okay, so carbonaceous BOD is typically something that we can measure, and usually this is done by empirical measurement, whereas nitrogen BOD, there are all these sort of, nitrogenous BOD often just happens very, very quickly, okay? If you think about carbon to nitrogen ratios with plants, for instance, you know, you've got a lot of nitrogen that gets consumed really quickly, and you can't really measure it in 1 or 2 hours or so, or you know, in a 1 or 2 day process, okay? So what happens in something like this is often we would measure nitrogenous BOD as a constant and just say, okay, we're going to subtract this amount of nitrogenous BOD, and possibly we do a couple of tests for that just to see how much oxygen it is. But when we're trying to calculate these overall rates, we often actually kill the nitrogen reaction, and we do that with a little. Where's that little sucker? I can't see that now. Hang on, sorry. We actually inhibit that with something called TCMP. So nitrogenous BOD, when we're trying to measure it, we inhibit it with something called TCMP, and we kill those the nitrobacter and all of those things in. So that's kind of the basic. How do we actually measure biochemical oxygen demand? Well, it's not really that difficult. What we'll do is we will take a sample of the effluent. So we go take the sample of the effluent, and we also take a sample of the receiving water body. So the effluent might be, you know, from that sewage plant, okay, and the receiving water body. And we're trying to measure it, you know. Typically what we do is something called a BOD5. We're looking at about a 5-day BOD, and once we know that, it's fairly predictable about how, you know, the rest of that goes and works. So we take that effluent, and we're going to mix the effluent with water from the receiving water body. Okay, so we will dilute here. We're going to dilute with receiving water body, RCW, okay, or RWB. Why do we want to dilute with water from the receiving water body instead of sending our grad student over to the faucet and just putting water from our Columbia City water?
Make sure you have similar bacteria that are present in the receiving waters.
That's right. What happens if you had to put Columbia water in there? You wouldn't have the sort of reaction, you know, you wouldn't have that biochemical oxygen demand going on, because in Columbia City water, it's almost devoid of those bacteria. Now the bacteria would probably be sitting in the effluent, but the main thing is you put the receiving water body, because that's a representative population of bacteria that are going to break it down. So that's going to give you a better idea of what that's going to look like. Now we're going to add TCMP. What does TCMP do?
Stops the nitrobacter from metabolizing.
Correct. So it stops anything to do with nitrogen breakdown, but the breakdown of that carbon continues. We are going to then aerate that. And I think it's at a specific temperature. It's 20 degrees C. So we want to keep it at 20 degrees C. So you aerate it until that sample is saturated. Okay? And then what you do is you have, now this is a 500 milliliter bottle here. And you have a little probe in here that measures the amount of oxygen in your in your bottle. Now, what happens in the beginning, especially when you have a very high BOD concentration? What would you imagine happens very quickly over here with the amount of oxygen you have in here?
Depleted.
It depletes. It gets eaten up like 2 or 3 hours, and man, it's down. So what you don't want to happen is now let's have a look. You don't want your DO to go below 2 milligrams per liter, because if that happens, your bacteria are all aerobic. Below 2 milligrams per liter, you're going to harm your bacteria population. So what you actually do is you find out how long it takes, especially in the beginning, for that. And typically, in most guys are not going to come back at 12 o'clock at night to re-aerate. So they're going to figure out how much I need to dilute so that I can re-aerate, say, tomorrow morning. So basically what's going to happen is, if you look at a graph over time of O2 in the solution, you're going to look at a graph over time of O2 doing this. Okay? And there's 2 milligrams per liter. And the smart lab guy is going to, before it gets to 2 milligrams per liter, he's going to go ahead and put oxygen down here. He's going to re-aerate it back up to, I think 7 milligrams per liter is about saturation at 20 degrees C. And then what happens is, what do you think? Do you think this is going to take a little bit longer to get down to that point this time? If I say it did, why would it do that?
There's less to metabolize.
There's less they used the word. I was battling for a word. There's less organic matter. There's less organic waste to metabolize. So over time, you can aerate it, and then eventually, you aerate it. Okay. So how do you measure your BOD? Well, you measure the amount of oxygen that's consumed from here to here, but then you add this, right? Because you added that oxygen there, right? You add this oxygen here. So let's say we were at 7 and we went down to 2.5. And we added 5.5. And we went down to 3 and we added 4. We went down to 4 and we added 3. And we ended up over here at, say, 5 milligrams per liter. Okay? What's my BOD? Well, my BOD would be 7 minus 2 equals 5 milligrams per liter plus 5.5, right? Because I added that over here. It was consumed plus 4 plus 3 plus, you started off at 7, and after 5 days, this is 5 days, I've got 5 milliliters. I started at 7, so 7 minus 2 equals, 7 minus 5 equals 2 milligrams per liter. So it's going to be 5 plus 5.5 plus 4 plus 3 plus 2. So 10.5, 14.5, 17.5 equals 19.5 milligram per liter. Does that make sense? Because we're watching how much of this is being consumed at the time.
That last number is the change from the beginning to the end?
Correct. You're looking at it from there to there.
Okay.
Now,
that quantity is still measured in milligrams per liter. It's not like moles of oxygen or grams of oxygen.
Correct. Yeah, because in this case, we're not worried about whether, what the composition of the stuff is. We're only worried about oxygen because we're saying that's the, especially for effluent calculations, that's the thing that we're measuring the most. And now, buried in some deep notes, and I used to do this for you guys, was I had a spreadsheet that, I don't know, maybe it's here. Okay, I got rid of it. But actually there are formulae for you then to be able to back calculate all this, because it's also based on your dilution. So it's just kind of formulae, what's the ratio of my dilution, how many times did I re-aerate from what point to what point. So there's some details in that, but what I wanted to do was give you the conceptual idea of what biochemical oxygen demand is. Okay, we just want to know how much oxygen is there. Why? Because fish don't know how to hold their breath for more than a minute, and that's kind of the issue. And so this particular biochemical oxygen demand has been around for a heck of a long time. And there's a model called the Streeter-Phelps model. And if any one of you wants to do the Streeter-Phelps model, it's kind of cool. The Streeter-Phelps model is kind of a cool way to model if you wanted to make your own with an Excel spreadsheet. It's kind of cool, it's satisfying, because there are very few parts, there are not that many moving parts in it, but you can create these cool graphs of it. And the Streeter-Phelps model essentially looks at what happens to oxygen in the water column. Okay, and there's a piece of software called Qual2K or Qual2E that actually models that. And so basically, you're looking at sediment oxygen demand, and then you're looking at demand. I can't read this. Let me go to the other one. You're looking at, if this is the surface of the water, and aquatic DO, you can calculate from that the amount of oxygen that diffuses into the water based on the depth, based on the rapidity of the motion, wave action, and that kind of stuff. You can also calculate how much oxygen is being removed from that water body, that's going up. You can calculate then also from oxygen going in from plants, from aquatic plants. But at night, what do? Aquatic plants breathe. So you can also calculate that. You can calculate how much oxygen fish use, benthic inhabitants use. So that is usually removal of oxygen from that system. You can also calculate how much is, so critters, I'm going to say critters, and I'm going to say organic. Let's call it organic matter rather than organic waste. So organic matter or organic waste is also going to use it. And if you've got effluent, that's going to be your biggie in that calculation. And this, I'm just using this Streeter-Phelps model thing over here. And then you've also got something called sediment, SOD, sediment oxygen demand. Okay, so that's stuff in your sediment. Let me ask you, in the sediment oxygen demand, would you say that there was more demand if you had this thick, gooey sludge that looked like the pluff mud down at? Well, in fact, let's look at pluff mud. Let's compare in your hometown.
When you were talking about that earlier, that's what I was picturing.
That's right. So if you consider pluff mud and you think about the sediment oxygen demand if the pluff mud was covered by, say, 2 foot of water, and then compare the sediment oxygen demand of the same area and the same amount of water over, say, a beautiful sandy beach, which would have the highest sediment oxygen demand, and why? I'm going to put you on the spot since I used your town.
The pluff mud?
Pluff mud, why?
Because I guess all the sediment, I don't know, because of all the sediment or what the organic would do.
bunch, it's just like my kids say, there's a buttload of organic material in there. I mean, it's boatloads of organic material, and that's the stuff there. What's that beautiful ocean beach got? Those minerals, those sands. Is there much?
No, I mean, it's just,
Yeah, it's just minerals. It's just silica. There's very little organic matter. So it stands to reason that if you've got this black sludge down there, you know that when you put it underwater, it's going to suck that oxygen out there because there's all this biological activity going on. Very little biological activity that's going to go on in the mineral sand. Okay? And so if you look, I think the next page, oh, here, SOD, and you can see some of the, in that second last slide, some of the sediment oxygen demand that goes on over there. I want to also just kind of point you to the aeration coefficients because I've been known to ask exam questions. But if you have a look at the aeration coefficients, you will see that depth of water and velocity of water have an influence. So let me ask you, if I have a stream that's traveling at 5 miles per hour and is 5 foot deep, and I have a stream that travels at half a mile per hour and that's 5 foot deep, which one would I expect to have more aeration going on?
Faster one.
The faster one, yeah, because you've got that turbulence, that you've got the motion that's trying to suck that water in. If I have a stream that's going at 5 miles per hour, it's 5 foot deep, and I have another stream going at 5 miles per hour and it's 20 foot deep. Where can I expect more aeration in that water body?
Shallower? Probably the shallower.
It's going to be the shallower one. So you can see over here, in these, you've got, for instance, you've got the O'Connor-Dobbins, which is for a certain regime of depth and a certain regime of flow rate. The Churchill and then the Owens-Gibbs equations. So these are just re-aeration coefficients of researchers that have found under certain conditions. And being able to do this, then we can calculate how much oxygen has been consumed, therefore also the rate of oxygen consumption in that water body, and how far or how long it would go, or what the lowest, the big idea would be what is the lowest amount of oxygen concentration in that water body for a given amount or rate of effluent and a given rate of flow. And so that's where the SAG, the modeling, the BOD modeling comes in. That last thing over there is that once you have a handle on your Streeter-Phelps model, then you can basically say oxygen on this side, and this is time and or distance, all right? So at this point, my oxygen is over here. Now I add my effluent, and basically what happens is, if once I add my effluent, what's going to happen to the amount of oxygen in the water? Will it go up or down just after the effluent?
Down.
It's going to go down because it's consuming. Right, so while the oxygen is being consumed, what's happening to the amount of organic matter in the effluent? That's also being consumed, right, by the bacteria. And not only is that happening, but we've also got, for instance, oxygen coming into that water body and replenishing. So this is not just the rate of oxygen consumption. This also represents the re-aeration coefficients and SOD and everything. And you get to a point in this process where the amount of oxygen being consumed is equal to the amount of oxygen being brought into that water body. And you kind of get to a kind of a turning point in that equation and then very slowly you find that you've got less and less and less effluent. Over time and over distance that you have to deal with, okay? And so this is kind of your critical point. If your criterion for fresh waters is 4 milligram per liter, you don't want that turning point to be less than 4 milligram per liter. All right? So you want it to be here, 4 milligram per liter. And you usually, your calculated flow rate that you take is usually kind of a low flow. It's called 7Q10. It's the lowest 7 days of flow in 10 years. And it's kind of a statistical calculation, 7Q10. And I swear I don't know how to calculate 7Q10 to save my life. But basically you don't want to calculate it on the day that it's raining cats and dogs. You want to calculate it on a very low flow, and you want to calculate it for summer or winter conditions. You want to calculate it for the worst case scenario where and that's typically going to be in the summer, because that's kind of your, what we call critical conditions, okay? So that's basically it. And I've got notes and all that, where you can actually mathematically calculate this with ordinary differential equations, or you can just be like me, because I did second year math, but I didn't really do well in it. And so, but I love crunching the numbers, and you can crunch those numbers in an Excel spreadsheet. So if any one of you is interested in doing a Streeter-Phelps model for your final class project, I'd be happy to help you set that up. Okay, so any questions on that? I'm going to close this